Thank you very much for that very kind introduction. And thank you to the SID for giving me the honor of delivering this year's Eugene Farber lecture. Those are my conflicts of interest. So I was lucky to meet Eugene Farber on one occasion when I was a fellow at the University of Michigan. And I think it's true to say that Eugene Farber was one of the preeminent dermatologists of the 20th century, not just in this country, but also internationally. He was the chair of the Department of Dermatology at Stanford for 36 years and was president of this society. He's best known, though, around the world for his pioneering and visionary work in psoriasis, the founding of the Psoriasis Research Institute in Palo Alto, and also the establishment in 1971 of the International Psoriasis Symposium. And reading his work and reading about him, it becomes very apparent that he was a man ahead of his time, that he was practicing translational science even 50, 60 years ago, and that he understood the importance of integrating clinical observation with investigative research. Now, when I was a boy, one of the books that I read was this book, because most of us clinician scientists are actually naturalists or amateur naturalists. We are chroniclers and describers. And Gilbert White was a country vicar, a country parson in rural southern England, in Selborne. And over the latter part of the 18th century, he chronicled very accurately the wildlife that he saw in that area. He didn't always understand what it meant, but he was able to write it down and form hypotheses. And a good example of that is that he knew that swallows arrived in the summer and disappeared in the winter. And his hypothesis was that they hibernated at the bottom of ponds, as do frogs and toads, because at that time migration was unknown. But he'd formed a hypothesis, and that's what we do as clinician scientists. We might not be able to answer it, but we can come up with the observations. So Eugene Farber wrote about variations in the natural history of psoriasis and very accurately described situations which we still see and which we still can't explain. He described the appearance of psoriasis at times of stress. He described that weight gain was important in development of psoriasis in predisposed individuals. And he also described patients with guttate psoriasis where the psoriasis disappeared and never returned. These are important and salient observations which direct the work that we should do. So this is the year of translational research. The GIID is devoted to that over this year, and this eloquent editorial written at the beginning of the year by Barbara Gilchrist, the editor, she describes the translational research process. And it's not just simply clinical observation and laboratory research melded together. It's a big team approach, and also it's the involvement of industry and how industry are key partners as we seek to find the best way to manage our patients. So this is going to be something of a philosophical discussion, and I'm not so presumptuous as to say this is the natural history, which is why I've called it a natural history. I'm going to introduce you to endotypes, the importance of whole patient research, what we have in the UK to approach investigating and understanding psoriasis, and then introduce you to the holy grail, if you like, of all of this work, which is stratified medicine. Now, those of you who have been around the SID for a few years will not have failed to see that psoriasis has gone through a fantastic time over the last 10 years. We know more about the disease and have better treatments than we've ever done before. And that's even come to the attention of Nature, who had a supplement devoted to psoriasis about 3 years ago. But psoriasis is about the whole patient. It's not just about a single cell, and it's not just about a single signaling. It's about looking at the whole patient. Looking at this picture, this painting, you say, well, this is a landscape. These are hills. It might be the Alps. It might be the Grand Canyon. It might be the Dolomites. We don't know what it is. And I'll come back to this and the relevance to what I'm talking about at the very end. Now, it's always important to read outside your specialty. Respiratory medicine have been there ahead of us in this regard. You could take out the word asthma and put in psoriasis, and it's heterogeneous, complex disease. That's exactly what psoriasis is. But what the respiratory physicians did, they came up with the idea of endotyping. And endotype, which is a shortened form of endophenotype, are subtypes of disease which are defined functionally and pathologically by a molecular mechanism or by treatment response. And that's what we're trying to do in psoriasis. Now, Robert Willen, who was an exact contemporary of Gilbert White, whilst Gilbert White was down in Hampshire describing the wildlife, he was up in London describing his psoriasis patients. He described the morphology and he differentiated psoriasis from leprosy, but also he described the patients. He understood that these patients were melancholic, they were depressed, they had psychosocial disability. So he again was ahead of his time and thinking about the whole patient. And that brings us to the first sort of big collaboration that I'm going to talk to you about, and that's IMPACT, which you've just heard about. IMPACT is funded by the National Institute for Health Research in Manchester. It's called the Identification and Management of Psoriasis-Associated Comorbidity. And these are not basic scientists. These are people, these are psychologists. These are health economics. These are statisticians, GPs, working together to understand the whole patient with psoriasis in primary care and how we can identify the comorbidities and manage them better. And about 2 years ago, we wrote an editorial on this, where we recognized that psoriasis is a chronic, lifelong disease. It is frequently life-ruining. It has very significant psychological and physical and social impact. And to research it and to manage it as a simple skin disease is not doing our patients justice, and is not doing ourselves as clinicians justice. In fact, this meeting is really dedicated to quantitative research, but qualitative research plays a very important role, because interviewing patients and interviewing those who look after them gives us the questions that we as scientists must be trying to address, questions that arise from direct patient care. Now, psoriasis nowadays is known to have a number of comorbidities. It's thought of as being a systemic disease. The links with psoriatic arthritis, the links with Crohn's disease, the links with cardiovascular disease. I'm not going to get into that potential minefield of whether psoriasis is an independent risk factor or not, and also the psychosocial burden. But there are important parallels. Certainly in cardiovascular disease, we know, in fact, there's an abstract I've seen here at this meeting from Michigan. We know that the inflammatory pathways we see in a plaque of psoriasis are very similar to those that we see in a plaque of atheroma. And you might ask, well, was that relevant actually to the psychosocial burden? Well, patients with psoriasis, about a third of them are depressed or anxious, just as Robert Willen described. 1 in 10 may have actually had significant thoughts of suicide, and 1 of 5 have experienced stigmatization and rejection. And the question is whether that could be part of that systemic inflammatory response as well. And this is an interesting study. This is a review, it's a nice title if nothing else, but it's a very good review on the effects of peripheral inflammation, arthritis in rodents being associated with neuroinflammation. Cytokines sing the blues. Inflammatory cytokines in peripheral inflammation can produce neuroinflammation, which leads to detachment of serotonin or 5-HT from its receptors and leads to depression. Can that be the case in psoriasis? In Manchester, we've done a lot of scanning studies in our patients, and we have a very sophisticated PET scan set up. And what we've been able to show, that those patients with psoriasis, using PET scan, those who are depressed have neuroinflammation. As you can see here, this red and orange in the brain of the psoriasis patient indicates inflammation. And that, in a way, answers a question or an observation we make in clinical practice. We put a patient with psoriasis on one of these powerful biologic therapies. They come back and see us in about 3 or 4 weeks' time. Their psoriasis might not have improved very much, but they feel much better. They're almost euphoric. What is happening is that probably you're reducing the neuroinflammation. The serotonin is now repopulating its receptors, and in essence, the biologic is the world's most expensive Prozac. But that's what's happening. Most of the research money, as you know, in psoriasis at present has been spent on understanding the very complex genetics of the condition. There are at least 41 psoriasis susceptibility loci in Caucasians alone, but the standout constant factor is that early onset psoriasis is associated incontrovertibly in about 60% of patients with HLA-CW6. Now, that has some important connotations. I introduce you to one of those, which is around late onset and early onset psoriasis, something we have an interest in. Most cases of psoriasis, 75%, occur for the first time before the age of 40, in fact, before the age of 35. There's a smaller subset where it occurs for the first time, usually in the 50s and 60s, first described actually by N.O. Christophers. HLA-CW6 is associated with early but not late onset. Single nucleotide polymorphisms and IL-1 beta are associated with late onset psoriasis but not early onset. And epidermal Langerhans cell migration in uninvolved skin, which we have a big interest in Manchester, which doesn't, Langerhans cell migration in psoriasis does not respond to either IL-1 beta or TNF alpha in early onset, but in late onset, it does respond to IL-1 beta. Again, a difference. And the final difference is that in just straight histology, you can see that in the epidermis, there are more CD8 T cells than CD4 in early onset, and it's reversed. There's more CD4 T cells in late onset compared with early onset. So we are convinced that these are different diseases, and that has relevance to the way that we manage them. And most studies don't focus on late onset at all. They're all on early onset disease. So when people go back to their clinics, you'll be aware that there's 41 psoriasis susceptibility loci. That means that these patients with chronic plaque disease actually have different conditions. There's probably different forms of the disease sitting there in the clinic on a Monday morning. We just need to be able to match that up to the genotype. When I started dermatology 32 years ago, I was very lucky to work with Lionel Fry at St. Mary's. And Lionel Fry, those of you who know him, is something of an iconoclast. He pushes back on dogma, and he was convinced that psoriasis was an immune-mediated condition. And he and Helgi Valdemarsson, an immunologist, put together the T-cell-mediated hypothesis in the early 1980s. That didn't really get a lot of traction with the scientific community, but what really made people sit up and notice was when it was shown, ourselves and the University of Michigan and others, showed that cyclosporine, which is a rudimentary T-cell targeted therapy, was highly effective for psoriasis. And that sort of indicated that probably T-cells were important in the disease. And at the same time, Sandoz, as they were, Novartis as they are now, started to invest in understanding the immunology of the condition, which has now led inexorably to the biologic era. And along the way, other important studies, particularly from Jim Kruger and Alice Gottlieb, this study particularly, where they used a fusion protein of diphtheria toxin and IL-2, which binds only to IL-2 receptor-expressing cells, T cells, and not keratinocytes, works for psoriasis. Again, further evidence that it's a T-cell-mediated disease. And with that shifting timeline, we're now at the stage where psoriasis is very much thought to be an IL-17-driven disease. In fact, one could say that psoriasis is the prototypic IL-17 disease. Not only is IL-17 produced by Th17 T cells downstream from IL-23 and TNF-alpha, but importantly, it's also produced by mast cells and neutrophils. And the proof is by targeting interleukin-17. And targeting of interleukin-17, of which there are 3 approaches, targeting interleukin-17A with secukinumab or ixekizumab, or targeting the receptor with brodalumab, produces the most impressive results from any treatment we have used so far in the management of psoriasis. I'm just going to give you examples from one of those biologics, one of those anti-IL-17s, and that's secukinumab, because it's the first one that's been approved for the management of psoriasis. And what we see with this is that patients actually achieve what's called a PASI 90, a 90% improvement in their psoriasis, or even a 100% improvement, a PASI 100, in 70% and 40% of the time. These are far outstrip any of the other data we've had so far. Not all patients respond, and some patients lose response, but that underscores the importance of IL-17. So we're now at the biologic era for psoriasis, and this has been transformational for our patients with severe disease. But there's variability in response, there's toxicity in some patients, and some patients have secondary failure. They respond and then they lose response, probably due to the development of anti-drug antibodies. So that sets the scene for understanding why that happens. And in the UK, we are interested in the safety of biologic therapies and systemic therapies. And we set up something called Bad Beer. That's not a poorly performing microbrewery. It's the British Association of Dermatologists Biologic Interventions Registry. That was set up in 2007. It's a national web-based safety register. Patients are not just biologic therapy, but conventional systemic therapies. We have 152 dermatology centres around the UK and Ireland who are contributing to this. We've got very rich, powerful phenotypic and quantitative disease severity data at baseline, and also response to therapy, quantitative response to therapy. And importantly, at least 30% of those patients, increasing all the time, have serum samples, DNA, and RNA. So it's a very powerful biological resource. And the current state of play, as of yesterday, is we have over 10,000 patients in this registry. But the important thing is that those 152 dermatology centres, many of them, they'd never done any research before. They're excited about being part of a big research program, and they're now starting to see these papers being produced, and they want to do more research. So that has inspired people such as Nick Reynolds and others to set up, based on the success of Bad Beer, something called UK Trend, the UK Translational Research Network in Dermatology, which is going to harness the power of all those dermatology centres who want to do translational research. So this brings us to stratified medicine. And of course, there's nothing ever new in medicine. Someone has thought of it before, and usually Hippocrates has got there before us 2,000 years ago. So this is Hippocrates' take on personalised medicine. It's far more important to know what person the disease has than what disease the person has. And stratified medicine, or personalized medicine, or precision medicine, or P4 medicine, whatever you want to call it, is if all of you in this room have psoriasis and I gave you a Etanercept, I would not know just by looking at you who was going to respond well, who was going to respond poorly, who was not going to respond at all, and who was going to have side effects. And what stratified medicine does is take the guesswork out of it and make it much more targeted to the individual. And David Cameron, he might not be Prime Minister tomorrow, but he certainly has helped us in stratified medicine, because in 2011 he undertook to put over close to £100 million into stratified medicine. And the reason that they did that was that they could see the global market opportunity for stratified medicine, the success of the treatments that have been used for breast cancer. In oncology, companion diagnostics are almost de rigueur. Herceptin is a good example. Given to women who have breast cancer that expresses the EGFR receptor, you have a companion diagnostic. Barack Obama has also seen the light. Earlier this year announced over $200 million for stratified or personalized medicine. And in the UK, it's a great opportunity, because whatever you might say about the National Health Service, it is a fantastically powerful collaborative testbed for this sort of approach. The 100,000 Genome project is to genome sequence 100,000 patients in NHS, and we can use those 60 million patients in the NHS for sort of stratified medicine platforms. It's a very powerful opportunity. Now, this isn't just around cancer. This is now starting to infiltrate into inflammatory disease. Again, the respiratory medicine doctors got in there ahead of us. Lebrikizumab is an anti-IL-13. When they did the first studies for asthma, the results were somewhat disappointing. It didn't seem to work too well. But when they stratified for those patients who had high levels of periostin, produced by bronchial epithelial cells in the blood, there's a much greater response. So this drug is now used with a companion diagnostic for periostin. High levels, you use the drug. Low levels, you don't. We're already seeing this sort of response in our clinics. Patients on adalimumab, an anti-TNF approach, it doesn't work so well in those patients who have nail disease. So that's the stratification approach. So the Medical Research Council were tasked with finding diseases which were stratifiable. And they wanted them to be consortium-based and collaborative, and the industry had to be engaged from the outset. And a group of us in the UK put together the hypothesis around getting psoriasis as one of those 12 that were funded, and it was funded, using clinical genetic and immune biomarkers to predict and reproducibly stratify the response of psoriasis to biologic therapies in a way that's minimally dosed, minimally effective dose, to produce algorithms or stratifiers which will be scalable for clinical use, but ultimately to produce healthcare saving. So this is the SORT consortium, Psoriasis Stratification to Optimize Relevant Therapy. It's not just academic partners. These are not just clinicians and basic scientists. These are systems biologists, they're machine learners, they're bioinformaticians, statisticians, working also with industry. You'll note there are 10 industry partners here, 7 pharma industry, and 3 diagnostics, if we do come up with a stratifier. And this isn't just a simple financial or fiscal transaction. This is collaboration in kind. Industry have data sets, industry have material, industry have expertise in bioinformatics. But also, we're working with the patients, the patient group. And the idea is to use the right biologic first time for these patients. We have 2 work strands. First one, this is led by Catherine Smith in London and Richard Warren in Manchester. It's looking at those endotypes associated with treatment outcome, the drug levels and anti-drug antibodies. How do they track the secondary failure? We're developing more sophisticated sensitive assays which can work in a drug-tolerant environment. But also, we're assessing adherence. The main reason that patients fail treatment is that they don't take the medicine as prescribed. I'm willing to wager that none of you in this room, after all, we're all patients, have taken a course of antibiotics as your GP has told you to. That's non-adherence. The second work strand is led by Frank Nestle in London and Nick Reynolds in Newcastle. And this is looking at transcriptomics mainly, in skin and blood, serial biopsies at baseline, 1 week, and 12 weeks after starting a biologic therapy. We're also looking though at metabolomics in the urine, and we're looking at the microbiome. And these are integrated work streams. They're not standalone. First of all, we're going to look at ustekinumab and secukinumab. And Novartis have embedded a SORT trial within their open study of secukinumab for TNF-alpha inhibitor failures, and also going to look at adalimumab. And that goes through the classic discovery, refinement, and validation process, and validation of the Badbeer resource. We're going to look at drug endotypes and disease endotypes. So drug response and disease endotypes, we're stratifying the disease and coming up with a new taxonomy for psoriasis. And why is this so important to industry? Well, that taxonomic classification based on mechanisms will allow us to identify cohorts of patients well suited to proof of principle testing of new molecules, much more cost-effective and cost-efficient. Stratification tools, if we're able to develop those, will facilitate accurate targeting of new molecules to patient or drug endotype. Also, they can establish minimal effective dose. Some patients don't need to be dosed as frequently as they are. And also, psoriasis is ahead of the game. We're using anti-IL-17 and anti-IL-23 ahead of other diseases, and we can inform arthritis and we can inform inflammatory bowel disease. So we've sort of gone through a proof of concept. Jim Kruger has done a lot of this proof of concept. But we wanted to see if this would work using etanercept. And this was a pilot study which was ahead of the SORT program. We call it pre-SORT. It's based in Manchester. It's by one of our research fellows, Amy Fowkes, and bringing together the expertise at GSK and various academic institutions to see whether we could do this in a small cohort of patients on etanercept. We had 10 patients, those serial biopsies, and as it worked out, it's just as we would predict. 1 failed, 3 did very well, and 6 didn't do quite so well. And the sort of data that she's getting is what we were hoping for, is that there was a difference in the transcripts, those who were responders and the non-responders. So you can see how this is certainly setting the groundwork that this might actually work. Now, there is data that this might work. There's one small study from Italy, this one, and one from Taiwan, which show the same thing. HLA-CW6 predicts response to ustekinumab. So if the patients are CW6 positive, they're 90% chance of getting a PASI 75. If they're negative, there's only a 65% chance. Some studies we've done in Manchester, working with Pfizer, going through their previous data sets, have shown that patients who have early onset disease are more likely to respond well to etanercept than those who have late onset psoriasis. So to close, part of that impact program, one of the work streams was looking at the incidence and prevalence of psoriasis. We did a systematic review, which was published in the JID about 3 years ago. And we showed, as most people have before, that psoriasis is more prevalent in Northern Europe than in Southern Europe. But also we found that there was some evidence to suggest an increasing trend of incidence of psoriasis over the last 30 years. Now, that could be because there's better data collection. It could be that more patients are coming forward for treatment. Now we have better treatments. Or it could be a genuine increase. So there is a need for prospective studies on the determinants of the natural history of psoriasis, both the remission and flares. We've been negligent. Eugene Farber was telling us to do this 60 years ago. We should know what the natural history of psoriasis is. What happens when a child presents with psoriasis? What's going to be the natural history? Also need a global study of the current disease incidence and prevalence. And that gives us a huge opportunity. Something called the Global Psoriasis Atlas, which is a joint initiative between the International Psoriasis Council, the International League of Dermatological Societies, and the International Federation of Psoriasis Associations, based on a very successful atlas developed in diabetes 14 years ago. You should go onto their website and have a look at that. This is what we want to try and do in psoriasis. We'd start off by redoing that systematic review, but look at using the grey data as well, and then developing methodology that we can use with local nuances in any country around the world. So we'll be able to tell you what the prevalence of psoriasis is in Brazil, and Bermuda, and Bahrain. So to close, I've sort of given you a philosophical overview of a natural history of psoriasis. It's complex. And the new taxonomy, which is what we must undertake, will depend on those endotypes. I've given you an example of how late onset and early onset psoriasis can be separated, which has important consequences for treatment. The role of comorbidities in this, particularly the psychosocial and the cardiovascular. If we get it right, that sort stratification process can lead to accurate, effective, and economic prescribing, the so-called right biologic first time. But amongst all of this, remember that we're treating a very complicated organism, which is a person, and it's not just the skin. And I'll take you back to that picture I showed you. This is a landscape. This is analogous to looking at a single cell type in psoriasis, or looking at one cell signaling pathway. This misses the whole picture, because the whole picture is this. This is psoriasis, this is the person. And if you just concentrated on that one cell type, you'd have missed it. So I'd like to thank you for your attention, and these are the people that I'd like to thank, particularly current people working with us on psoriasis, and three of my mentors, Lionel Fry, John Voorhees, and Brian Nickloff. Thank you.